In the thoracic surgery cancer biology lab, our goal is really to merge the basic science aspects with the clinical aspects. We are studying both lung and esophageal cancers. These are cancers that unfortunately have very poor prognosis, about 20% 5-year survival rates. And although we've made some headway surgically and in terms of therapeutic options, it's been slow in the past 2 decades. So we really feel like this is an area we need to focus on and come up with improved strategies for both the prevention and treatment of these cancers. To that end, we really take a team approach among our clinician scientists and basic scientists and also look at both the genetics, host factors, and environmental factors so we can better understand how these 3 groupings merge together to increase cancer rates, and therefore we can better try to figure out how we can target those for strategies and new therapeutic options to decrease risk for those cancers and or improve treatment outcomes. Some of the areas that we're focused on in terms of research in the laboratory include new biomarkers, including cell-free DNA. So when a tumor grows, it sheds DNA into the circulation, and we can measure that in patients and hope that it may be a new tool for looking at response to therapy or disease recurrence. And so one of our clinicians is working with a cross-institutional project on that. We're also looking at new chemopreventive agents, so can we decrease the risk of cancer progression in patients that have premalignancy, for example. And in my background, I'm very interested in natural products, food-derived and natural-derived agents, but we're also using some repurposed drugs that haven't looked efficacious perhaps for cancer prevention alone, but when you combine them with other agents, there's some synergy and you get low toxicity with good activity. And so we're studying some of those and specific effects on mitochondrial targeting agents as a new way to kill cancer cells. We have also investigators that are very interested in the microbiome, both in lung and myself in esophageal cancer, and how that may be used as a tool to increase therapeutic efficacy, or also could we give something in the diet that has favorable effects on the microbiome that might inhibit cancer progression. Finally, we're also interested in changes in the normal tissue. Something that's been ignored historically. So can we maybe change our diets or give something as a preventive agent that might have an impact on the normal epithelium, increasing its resiliency, so then when it's faced with a damaging agent such as reflux, so acid coming up from the stomach into the esophagus and damaging it, can we give something ahead of time that might help that tissue be more resilient and respond and repair better, for example? So ultimately our goal is to come up with new interventions and strategies that may benefit our patients and improve their prognosis, because both lung and esophageal cancer remain very deadly malignancies.